DNA structure. The DNA structure can be thought of as a twisted ladder. This structure is described as a double helix. It is a nucleic acid, and all nucleic acids are made up of nucleotides. The DNA molecule is composed of units called nucleotides, and each nucleotide is composed of 3 different components such as sugar, phosphate groups and nitrogen bases. The basic building blocks of DNA are nucleotides, which are composed of a sugar group, a phosphate group, and a nitrogen base. The sugar and phosphate groups link the nucleotides together to form each strand of DNA. Adenine, A, thymine, T, guanine, G, and cytosine, C, are 4 types of nitrogen bases. These 4 nitrogenous bases pair together in the following way: A with T, and C with G. These base pairs are essential for the DNA's double helix structure, which resembles a twisted ladder. The order of the nitrogenous bases determines the genetic code or the DNA's instructions. Among the 3 components of DNA structure, sugar is the one which forms the backbone of the DNA molecule. The nitrogenous bases of the opposite strands form hydrogen bonds, forming a ladder-like structure. The DNA molecule consists of 4 nitrogen bases, namely adenine, A, thymine, T, cytosine, C, and guanine, G, which ultimately form the structure of a nucleotide. The A and G are purines, and the C and P are pyrimidines. The 2 strands of DNA run in opposite directions. These strands are held together by the hydrogen bond that is present between the 2 complementary bases. The strands are helically twisted, where each strand forms a right-handed coil, and 10 nucleotides make up a single turn. The pitch of each helix is 3.4 nanometers. Hence, the distance between 2 consecutive base pairs, i.e., hydrogen-bonded bases of the opposite strands, is 0.34 nanometers. The DNA coils up, forming chromosomes, and each chromosome has a single molecule of DNA in it. Overall, human beings have around 23 pairs of chromosomes in the nucleus of cells. DNA also plays an essential role in the process of cell division. Chargaff's rule, Erwin Chargaff, a biochemist, discovered that the number of nitrogenous bases in the DNA was present in equal quantities. The amount of A is equal to T, whereas the amount of C is equal to G. A equals T, C equals G. In other words, the DNA of any cell from any organism should have a 1-to-1 ratio of purine and pyrimidine bases. DNA replication. DNA replication is an important process that occurs during cell division. It is also known as semi-conservative replication, during which DNA makes a copy of itself. DNA replication takes place in 3 stages. Step 1. Initiation. The replication of DNA begins at a point known as the origin of replication. The 2 DNA strands are separated by the DNA helicase. This forms the replication fork. Step 2. Elongation. DNA polymerase 3 reads the nucleotides on the template strand and makes a new strand by adding complementary nucleotides one after the other. For e.g., if it reads an adenine on the template strand, it will add a thymine on the complementary strand. While adding nucleotides to the lagging strand, gaps are formed between the strands. These gaps are known as Okazaki fragments. These gaps or nicks are sealed by ligase. Step 3. Termination. The termination sequence present opposite to the origin of replication terminates the replication process. The fuse protein, terminus utilization substance, binds to terminates sequence and halts DNA polymerase movement. DNA function. DNA is the genetic material which carries all the hereditary information. Genes are the small segments of DNA, consisting mostly of 250 to 2 million base pairs. Polypeptide chains are further folded in secondary, tertiary and quaternary structures to form different proteins. As every organism contains many genes in its DNA, different types of proteins can be formed. Proteins are the main functional and structural molecules in most organisms. Replication process, transferring the genetic information from one cell to its daughters and from one generation to the next, and equal distribution of DNA during the cell division. The functions of DNA include replication, gene expression, mutation, transcription and translation. What type of DNA is found in humans? B DNA is found in humans. It is a right-handed double helical structure.